As I stand here basking in the sun's embrace, breathing in the fresh air, it's easy to overlook the very causation of 10% of coral death: sunscreen.
Hi, I'm Shaanvhi Jayaram,
Licayan,
Rushil Kukreja,
Gabby, Licayan,
Shirdi, Palle,
and this is From Beach to Bleach.
a study where we tested how oxybenzone affects Aiptasia pallida.
Coral reefs, known for having the greatest biodiversity in the ocean, are significant ecosystems that provide support for marine life and benefits for humans. However, they face endangerment due to both the natural and human-induced causes, with around 60% of the world's coral reefs suffering from bleaching. One of the primary human-induced factors contributing to coral bleaching and mortality is the presence of oxybenzone, a harmful chemical found in sunscreen.
We performed this study because we want to test the effects of oxybenzone from sunscreen on the organism Aiptasia pallida. We specifically were looking for how it impacted movement, pedal laceration, oral disc size, and color. We collected data that helped us test our hypothesis, which was that increased amounts of oxybenzone would increase the reproduction rate of the Aiptasia because they tend to reproduce more when under stress. Alongside this, we also tested the aforementioned factors throughout the experiment. Oxybenzone, also known as benzophenone 3, is an organic compound that can be found in some sunscreens. Previous studies, such as one performed on G. vermiculophylla by Zing et al., showed that oxybenzone negatively impacted photosynthetic rate, pigmentation, and growth rate. In a research study performed by Vicovic et al., 21 anemones were tested, and 16 of them had been exposed to both oxybenzone and UV light died. This led us to also test this on another organism, Aiptasia pallida, a model organism. Using a model organism lets us apply the results of our study to other organisms like coral, which is most susceptible to bleaching. Coral reefs are ecosystems essential for biodiversity, marine life, and through the analysis of oxybenzone on Aiptasia pallida, which can model coral, a keystone species, we can further dive into the detrimental impacts of benzophenone 3 from recreational sunscreen.
Our study was on the effect of oxybenzone concentrations on Aiptasia pallida. Our independent variable was the oxybenzone concentration, and we had 3 levels, which were 0, 300, and 600 nanograms per milliliter of oxybenzone. We used 2 sunscreens, one oxybenzone-free Neutrogena sunscreen, and one containing 6% oxybenzone, which was the Copper Tone Kids sunscreen lotion. Each treatment group had the same amount of sunscreen, but different levels of oxybenzone due to the different types of sunscreens that we used. We had 6 replicates per group, and we had many constants in our experiment. Our dependent variables were changes in color, movement, pedal laceration, and oil disc size of the anemones. We measured these variables by using a color chart to measure changes in color. We counted the number of pedal lacerates that we saw, and we took pictures of the anemones under the graph paper each time we collected data to measure movement over time and the size of the oil discs. We collected data 3 times a week.
During this experiment, we conducted a study to investigate the impact of oxybenzone on the oral disc size, pedal laceration, movement, and color of Aiptasia pallida. The first graph displays the correlation between oxybenzone and the average oral disc size of Aiptasia. The 0 milliliter treatment group exhibited a 21% increase in size, while the 300 and 600 milliliter groups showed a 3% and 16% decrease in size, respectively. It is evident that all 3 groups experienced size reductions and increases on specific dates, such as March 28 to 29. Moreover, a one-way ANOVA test confirmed the same with a p-value of 0.05. This bar graph compares the average number of pedal lacerates per Aiptasia. We found no significant difference in the number of pedal lacerates among the 300 and 600 treatment groups. Nonetheless, we found significant difference in pedal laceration between the 0 and 600 treatment groups. The standard error bars on each indicates that the differences are insignificant. Additionally, a one-way ANOVA test confirmed the same with a p-value greater than 0.15. The third graph demonstrates the effects of oxybenzone on the average movement of Aiptasia pallida. We found no significant difference in the average amount of movement among the 3 treatment groups. Similarly, the treatment group movements had no particular trend. Furthermore, a one-way ANOVA test confirmed the same with a p-value well over 0.28. To demonstrate the effects of oxybenzone on the darkest visible color of Aiptasia, we used a heat graph. The data we collected found a statistically significant difference, as the anemones in the 600 milliliter treatment group had the most substantial decrease, while the anemones in the 0 milliliter treatment group had the slightest decrease. A one-way ANOVA indicated a statistically significant difference with a p-value of 0.03. Lastly, this heat graph displays the effects of oxybenzone on the lightest visible color of Aiptasia. Once again, we found a statistically significant difference as the anemones in the 600 milliliter treatment group had the most substantial decrease, while the anemones in the 0 milliliter treatment group had the slightest decline. A one-way ANOVA test indicated a statistically significant difference with a p-value of a little under 0.02. Overall, our research provides important insights into the effects of oxybenzone on Aiptasia pallida. We hope our findings can contribute onto the ongoing efforts to better understand the potential harm caused by oxybenzone and other harmful chemicals found in personal care products.
Since some of our data was significant, this indicates that oxybenzone negatively affects the oral disc size, pedal laceration, and coral bleaching of Aiptasia pallida. However, it is essential to acknowledge possible weaknesses and errors in our experiment. For example, we used only 6 replicates per treatment group, and the Aiptasia pallida used varied in size and age, affecting the accuracy of the data. Additionally, although the sunscreens used in the treatment groups were selected to minimize any potential confounding variables in active ingredients, the sunscreens still have different concentrations of homosalate, 13% for the oxybenzone-containing sunscreen and 10% for the oxybenzone-free sunscreen. This could affect the results of our experiment. Our study adds to previous research suggesting that oxybenzone has a negative impact on various physiological parameters of coral.
In the future, if we were to redo this experiment, we would increase the number of replicates to ensure a more stronger statistical analysis. By having a stronger sample size, we can better account for the potential variations and outliers, resulting in a more accurate and reliable findings. Moreover, it would be beneficial to expand our investigation to include other ingredients commonly found in sunscreens. By testing a variety of ingredients such as alpha-benzone, oxynitrate, and titanium dioxide, we can explore their individual and combined effects on Aiptasia. Additionally, conducting further experiments to explore different concentrations and exposure duration of oxybenzone would deepen our understanding of its specific effects on Aiptasia.
Throughout the study, we learned that one of the threats facing coral reefs around the world is the use of oxybenzone-containing sunscreens. Coral reefs are vital ecosystems, so protecting them from sunscreen pollution is essential for human well-being as well as environmental sustainability.
Individuals can reduce the damage caused by oxybenzone by avoiding using sunscreens that contain this compound, especially when visiting coral reef areas. There are alternative sunscreens that use mineral-based ingredients such as zinc oxide or titanium dioxide, which are less harmful to coral. Additionally, governments can ban oxybenzone-containing sunscreens in places where coral reefs are present to protect these fragile habitats.
Thank you, Dr. Morrow.
Ms. Harris, Ms. Iyengar,
Mr. Lightner,
And thank you everyone for listening.